Hi, everyone. Thank you for joining us for our weekly briefing on preventing and managing COVID-19 in the workplace. This is our 39th consecutive week. This is Karen O'Hara. I'm the Director of Marketing and Communications with WorkCare, your moderator for today's session. If you're a regular attendee, we're happy to have you back. And if you're just joining for the first time, you're in for a very interesting session because we're going to be talking about the vaccine. As always, this session is being recorded. You will be on mute during the webinar. Please use the Q&A function in Zoom to submit your questions. And it's a little easier for us to monitor the questions if you use Q&A instead of chat. We do watch both, but if it's consistently in Q&A, then we can keep track of them a little bit easier. The webinar typically lasts about 40 to 45 minutes. We reserve the last 10 to 15 minutes for questions. All questions are answered in writing as well and posted on our website. So those questions we don't get to are answered, and the ones that we do answer are also transcribed. We also post the recording on our YouTube channel, and Mackenzie posts those links during the session in the chat. I'd like to introduce you to our presenter, Dr. Anthony Harris. Dr. Harris is an MD, MBA, MPH. He's board certified in occupational medicine and WorkCare's Chief Innovation Officer. He's also our Associate Medical Director for On-Site Clinical Operations and our lead clinician for WorkCare's COVID-19 response. I know he has a lot to cover today, so I'm going to turn it right over to him. Thanks again for joining us.
Excellent. Thank you, Karen. And I want to do a quick microphone check here. Having some technical difficulties today. So can everyone hear me? Karen, can you hear me okay?
Yeah, if you fade in and out, I'll just give you a little sign.
Perfect.
So far, it sounds okay.
Excellent. So thanks everyone for joining us today. We're going to have some fun talking about vaccines, and we're going to talk about, obviously, the impact to our workforce regarding the vaccine that's coming to market. And we want to make sure that everyone is well aware of what the vaccines are and are not. We want to make you aware of some of the myths that are out there about the vaccine and some truths about vaccinations in general that may lead to our workforce not partaking in the vaccine once it's available. So without further ado, we're going to get right into it with the typical agenda for today, and we'll start with clinical update. So for our clinical update today, we are going to obviously focus in on vaccines, and in this case, we're going to talk about adverse reactions from vaccines. This is by far and away probably the most deepest concern that individuals have, both in the general public, also in the healthcare community, as well as our workforce regarding why should I take the vaccine if there's an untoward outcome possibility of receiving after I receive it. When we talk about the leading vaccines coming to market, everyone has likely heard of Pfizer. We've talked about it previously in our presentation. Moderna is following Pfizer with regard to their progression through their third or phase 3 clinical trials. And we spoke about Novavax previously. Admittedly, it was one of my top contenders several weeks ago. We're going to switch that out today with AstraZeneca because it looks like they're going to be the third to market with regard to their vaccine for COVID-19. So let's zero in on Pfizer's vaccine. Pfizer's vaccine, I don't have it on this slide, but we talked about it previously, is a mRNA vaccine, as well as the Moderna vaccine. The mRNA vaccine that Pfizer has put forth has several side effects that have been documented with those who are 16 and older. And the population that they looked at went from the 16 years all the way up to 55 and to older adults as well. And what we see in this huge population of individuals who were administered the vaccine, we're seeing from dose 1 to dose 2 a reaction in terms of adverse events within 30 minutes. Similarities between those individuals who received the vaccine, the title of the vaccine is BNT 162B2. And 78 out of 18,000, roughly almost 19,000 individuals had an immediate reaction in 30 minutes after receiving dose 1. As compared to those who received a placebo, 66 individuals out of 18,000, nearly 19,000, had similar reactions with regard to their administration of a placebo within 30 minutes after administration. Dose 2, that number is 52 from that population, almost 19,000, as well as those who received a placebo, 39 out of that 18,000. So we see these similarities, and that gives us assurance that this particular vaccine is not causing significant side effects early on after administration with regard to adverse events. If we look at then those who were solicited, meaning asked about a reaction within 7 days, what we see here is after dose 1, that number is 3,200 out of 4,000 that were solicited. If we're looking at dose 2, that number is 2,700 roughly out of roughly 4,000, 3,700. And if we flip over to the placebo, we're still seeing a significant number, 12.8%, 528 out of 4,000 that had solicited a response to their injection locally, particularly. So this is not a systemic effect. This is local effect only. Let's move on to solicitation for adverse events. After dose 1, 59% roughly who received the vaccine received a adverse event after dose 1. After dose 2, we see a similar 69.9%. It's less significant in those individuals who received the placebo, 47.0 and 33.8. Now let's continue to the individuals that have the risk of death, right? In this case, risk of death is something that's prominent amongst individuals in the general population, but the risk of death is 0 when we look at these populations of over 20,000, in this case, 21,000 individuals under this study. So we want to hold this in memory because it's important to be able to communicate to our workforce what the true risks are. Because again, there's a lot of myths out there around the risks, and there's a lot of myths out there around some things that we have seen and associated with vaccines in the past that don't necessarily hold true for the vaccines germane to COVID-19. So let's talk about now some pathology that is causing some fear amongst the general public. We're talking about allergies. We want to go into what allergies may exist to the vaccine, in this case, Pfizer, Moderna, as well as AstraZeneca, and should there be concern for an adverse reaction regarding allergy to these medications? The answer to that, as of yesterday, is yes. So we all know that the UK has received authorization, emergency use authorization equivalent in the UK for the Pfizer vaccine. And it began in the UK, where administration for their healthcare workers received authorization as well as monitoring for safety. And what we saw is that 2 individuals out of thousands that were administered the vaccine had an immediate reaction and an anaphylactic reaction to the vaccination itself. This is obviously the concern of many regarding, should I take the vaccine or not, realizing that individuals may have a reaction. So what is the response to individuals who have this fear? In the UK, the response was, If you have a history of previous significant allergy to vaccines, hold off taking the vaccination. Now, why was this not understood in this particular study, the phase 3 clinical trial with regard to Pfizer's vaccine of this type of response? And the answer to that is the vaccination protocols did not include, unfortunately, those who had had significant allergic responses to vaccines in the past. So when we fast forward to now and general administration to the general public, in this case, the healthcare workers, we see this now adverse response to the vaccine and subsequently the recommendation to hold off. So what is anaphylaxis? Let's get into that a little bit. We're talking about pathology. We're not going to make you physiology experts in this deep dive, but we want you to know enough to communicate to your workforce what it is at a high level, what causes it at a high level, and some of the things that should hopefully allay their concerns around vaccines, in this case, the COVID-19 vaccine. So some of the side effects that may happen with the vaccine, runny nose, the things that happen in the GI tract, such as diarrhea, vomiting, hives in the skin, can be all indications of anaphylaxis, early anaphylaxis. So when administered a vaccine, the protocol is to monitor each individual shortly thereafter for a period of time to make sure they're not having any of these signs or symptoms as a result of the vaccine administration. If we look at the mechanism behind these type of responses from an immunologic standpoint, it begins with the antigen. The antigen in this case is the vaccine itself. It's foreign to the body. The body then recognizes it through several pathways. We'll talk about the antigen interacting with B cells in this case. B cells are a type of white blood cell. They're important for us establishing immunity to foreign material, foreign bodies, and B cells then produce antibodies, right? In this case, IgE antibodies is what we're concerned about. These IgE antibodies then go on to attach to mast cells, and these mast cells release histamines that cause other immunologic responses to promulgate thereafter. So when we zero in on this pathway and we look at specifically anaphylactic reaction, the antigen, which causes excitation of B cells, then go on to attach and promote plasma cells to make a huge number of antibodies. And these antibodies are primed and ready to go when your body encounters antigens again, that same antigen again. And that massive release of antibodies, the IgE in this case, in conjunction with mast cells and releasing histamines, is what causes the anaphylactic reaction and the severe systemic effects that we all are familiar with and need immediate responsiveness to help these individuals. By the way, the two individuals who received the vaccine in the UK and had this response are recovering safely, so thankfully for that. So let's do the numbers. Let's look at the US. We're going to do this in an abbreviated fashion today. We all know how we're faring. We're not faring well in the US. So we will spare you the details on a macro level in terms of going through each of these type of maps. We'll focus in on the prevalence at this point, 15 million cases in the US. The average on a daily 7-day rolling average is 61.8. If we look at our total deaths, we're approaching 300,000 deaths. Some of the projections with regard to number of deaths by the end of the year is approaching 400,000 deaths, roughly. So we need to be cognizant to the realization that we haven't even peaked in regards to our impact from COVID-19. If we look at our trend, obviously the trend is continuing to go up in terms of daily new cases of COVID compared to where we were 2 weeks ago, where we were last week, and where we are today. we're still seeing dramatic increases in number of cases. If we look at this per million, we're still in the highest ranking position for impact from COVID per our population. That was 2 weeks ago, that was last week, and where we are today. Where we are today, obviously, France has overtaken us in terms of population impact, and we see their dramatic drop, overtook us previously, but see their dramatic drop in their per population cases. And we have to attribute that to the dramatic approaches that they've taken to hopefully social distance individuals appropriately. And it seems to have worked, right? So that gives us hope, but it also gives us an indication of how poorly we're adhering to some of those social distancing, PPE utilization, et cetera, to keep our population safe and reverse the trend that we're continuing to see in our rise. If we look at deaths, obviously we're in a similar position, with deaths being that 30-day lagging indicator, where we were 2 weeks ago, where we were last week, and where we are today. For our deaths per population, we were below France and the United Kingdom previously, started to escalate and continue to escalate, now in the lead position for per population number of deaths. And we'll continue to see that trend, unfortunately, after the Thanksgiving holiday and as we approach the Christmas season. Testing-wise, we saw a downward trend 2 weeks ago of testing. We continued to see that downward trend last week, and we're continuing to see that downward trend this week. So not boding well for our impact of testing interventions to help identify cases early and practice safe quarantining and isolation thereafter. So in cases in certain states, we know that we are woefully underestimating the true impact of COVID, and these metrics continue to point in that direction, particularly when we break it down per 1,000 individuals and where we existed 2 weeks ago, where we were last week, and where we are today. We're trending in the wrong direction. If we look at percentage of positive cases, that was trending in the wrong direction 2 weeks ago, continued to increase in that trend last week, and certainly in this case has still increased in that trend slightly. But again, pointing to the fact that we are not accurately articulating the burden of illness. If we look at other data, example, Illinois, 8 times what we see in the numbers today is the estimate for true prevalence of COVID-19 amongst the population. And this trend of underestimating true prevalence likely is across the board in multiple states. So as we talk about prevention and we relate it back to vaccines here, we want to focus in on opportunities to communicate effectively to our workforce. That is going to be key as we move forward with vaccinations, to get good information to our workforce, to the general public, around why they need to be vaccinated. So the opportunities to do so in terms of what to expect begins with FDA emergency use authorization of these vaccines. Pfizer is up tomorrow. So if anyone is interested in participating, it's open to the general public. We'll talk more about that shortly here. What we know to be true from the phase 3 clinical trials of Pfizer, and what we need to iterate to our general public and to our workforce, is that this vaccine is effective. When we look at efficaciousness of the Pfizer vaccine after dose 1, we're looking at an 82% effectiveness rate. If we look at individuals compared to placebo and those who received the vaccine, obviously we want to let people know that there is a difference, right? If we look at then those individuals who received the vaccine after dose 2, we see a 90.5% effectiveness. If we look at then after 7 days, that goes up to 94.8. So simply reiterating that after first dose, 82% of people have immunity. After the second dose, within days, 90.5%. After 7 days, 94.8%. The virtual meeting regarding the FDA's decision tomorrow, they've been meeting since the 8th of this week, but their decision tomorrow to authorize EUA for Pfizer's vaccine is public, available on YouTube. You can participate with us right along with the rest of the nation as we understand whether or not this is going to be reality to distribute the vaccine to our general workforce. As we look at the second contender, Moderna, their submission has moved forward. FDA has said on the 17th, they will consider Moderna's approval for emergency use authorization. If we look at other countries and what they've done with their vaccine efforts, China comes obviously to the forefront because they vaccinated nearly 1 million individuals in that country. And so we look to that to hopefully glean information and data around efficacy of vaccines, how long the vaccine provides immunity, and adverse reactions, right? Unfortunately, in this case, we have nothing. China has not released any clinical data even to the efficacy and adverse reactions to the vaccine itself. We know in terms of top contenders that China is followed by another vaccine within China that's already administered to the military there. But we want to look again at the top contenders, obviously Pfizer #1, Moderna #2, and AstraZeneca #3. And we anticipate EUA approvals of all three in the weeks to come. The EUA, in this case, regarding vaccinations in China that I was just speaking about, has released some information data on efficacy rates. 86% is where it landed in regards to the UAE, and their approach to trying to understand efficaciousness in the absence of that data from China directly. So we want to communicate again that vaccine campaigns are happening abroad. Hopefully we'll begin here in the US, and we're learning about safety. We're learning about efficaciousness now beyond the phase 3 clinical trials. And this is what we want to communicate to our workforce. So when we talk about messaging of COVID-19 safety of the vaccine, we want to look at data that has examined vaccines and messaging and issues around safety of vaccines historically. And if we turn to data from, in this case, a Deloitte report from 2019, we can see several things that still hold true today. One of those things around vaccinations that will hold us up and prevent us from being effective with distribution and administration is a sentiment from individuals and their perception around safety. And we see this both when the industry is surveyed, individuals within industry is surveyed, and we see this also in non-industry surveys of general publics. And this is a multinational body of work that was conducted last year. We also see something that we want to highlight, which is training and capacity for development and administration of vaccines. So when we talk about trying to understand the pain points amongst the community, we want to talk about how do we address it? How do we bring it to bear that we can convince our workforce? Obviously, it won't be compulsory as a means to get our individuals compliant with vaccinations, but we want to at least cajole them as much as possible that vaccines are going to be safe, are going to be efficacious, and are going to be a better pathway than going without. And if we look at leadership, we've seen this week, within the last several days, the previous presidents band together and will publicly receive vaccinations once available and approved by the FDA. We see that the misinformation that's out there can be effectively addressed. In this case, highlighting female fertility, perhaps one of the most widely proclaimed side effects of COVID-19 Pfizer vaccinations, the mRNA vaccination. The fear in general is that once the body produces antibodies from the mRNA, those antibodies will then attach themselves to the female uh, placenta and then cause fetal demise, right? And if we look at the data on the protein itself in the placenta, it simply is not true that antibodies produced from an mRNA vaccine from Pfizer or Moderna will have any efficaciousness in attaching to placenta proteins. And you'll see this, you'll hear this, you need to address it when you can amongst your workforce, because these will be things that prevent individuals from having confidence of getting vaccinated without significant adverse events. If we look at then some additional opportunities to correct misinformation or misunderstanding in the workforce, we know that workers will go Google the heck out of some of the available data on vaccines, and they will misapply oftentimes the data they find to generalize to all vaccines. What am I talking about? For example, in this case, if we look at toxoid vaccines, WHO has published data on safety and response from toxoid vaccine administration. In this case, at the bottom, you can see the immune response. It may require several doses and usually needs an adjuvant, right? That's information pertaining to toxoid. If we look at safety and stability, then we see information regarding safety as in terms of systemic reactions, very rare, local, and systemic reaction, usually stable for long-lasting periods of time. Now, if our workforce takes this information and generalizes it to COVID vaccines, not too much harm in that, right? Because there's not a significant profile of reactivity and adverse events for toxoid vaccines. But if we look at live attenuated vaccines and we go to what WHO has published around live attenuated vaccines and and if our workforce then generalizes to COVID-19 vaccines, we see that the safety profile for live attenuated vaccines can give some concern, right? That live attenuated pathogens can revert to their original form and cause disease, potential harm to individuals who have compromised immunity, right? So we're seeing bullet points from a reputable source that may be misinterpreted that causes our workforce to push back on getting vaccinated with COVID-19. And the list continues in regards to the potential for miscommunication, right? So we want to make sure that we're ahead of these concerns, we're ahead of the communications that need to happen now and hopefully has already started as we continue through this period of opening up vaccinations. And we want to then correct some of the things that we spoke to previously, or at least update, which is the deadline for receiving vaccines amongst the states and the impact that that's going to have to the timeline of administration to our workforce. If we look at the deadline that has already occurred earlier this month, we see that states are in position to receive boxes of the vaccine from Pfizer, roughly 972, I believe, doses in each box. And states are already estimating they're going to receive far less than what they originally anticipated. They will receive in the order of magnitude of 12,000, in some cases, more than tens of thousands of doses, but that's going to go to primarily the healthcare workers, police officers for the first phase, which is called 1A of vaccine administration. Ultimately, how does this impact our timeline that we published previously with our internal estimates based upon historic trends of vaccine administration here in the US? We see that the Pfizer vaccine has now will hopefully receive tomorrow its EUA approval. That's December, January. The states will then order their massive orders of vaccines. We're talking in the millions then, right? And that's going to occur late December, and then finally get to administration in beginning of January. So that's pushing it closer than what we said previously from February. The safety profile reporting will still continue through March and April. The mass order, again, for hundreds of millions will happen in May, preparing for general administration to our public, our workforce, and to the 1C category of distribution, which is our essential workers. Then when we talk about the potential for collaboration, we're still looking at May for WorkCare to receive vaccines for our general workforce. So this is the perspective we have now with regard to the timeline, and we're preparing for hopefully partnering with you all to provide this. If we look at one thing that we want to highlight, which is positive in regard to FDA approvals, FDA has now approved a COVID and flu combination test for home use. And this may have a significant impact on our absenteeism as we move forward. The Quest Diagnostics is the approval that was recently provided by FDA. And we want to look at opportunities to get that in the hands of our workforce to decrease absenteeism. So with that, covering the vaccine myths, we want to stop, ask questions, and we'll start that period now. Thank you so much.
Thank you, Dr. Harris. Could you give us a little tutorial on the mRNA vaccine technology and the history of it? Is it a relatively new, or is it something that's more experimental, and how does it work?
Karen, I cut out a little bit there. Say that one more time. I apologize.
That's okay. Just a tutorial on mRNA vaccine technology, the history of it, and how it works, just the basics.
Sure. I believe we covered that also in a previous presentation. And the mRNA vaccines, in short, are the building blocks for the antibodies that are produced by our immune system. And the mRNA enters the cell. It's then recognized and appropriately produces the antibodies that then exit the cell and cascade through the trend that I showed you earlier, through the presentation that I showed you earlier today. And that is true for both the Moderna and the Pfizer vaccine. The AstraZeneca vaccine itself is an adenovirus-produced vaccine, which is more classical in regards to how vaccines are produced. And we presented that data and that pathway also previously, where the proteins from the SARS-CoV-2 viron are extracted, placed into in vitro, placed into a culture that allows those proteins to be replicated. Sometimes it's replicated not in a adenovirus culture, and then those proteins are then introduced to individuals to promote antibody formation, in short, as I'm paraphrasing. So there is a difference between Moderna, Pfizer, and the AstraZeneca vaccine.
Okay, next question is about transporting the vaccine, which has to be shipped and stored under precise temperature guidelines, especially the Pfizer vaccine, which has to be super cold. How can we be certain that the vaccine hasn't been compromised due to higher temperatures for a length of time during the shipping process?
Sure. So the logistical records must be captured and are required for anyone who is distributing and administering the vaccine. It's called the cold chain, and manufacturers as well as distributors have to ensure through records that the vaccine has not been in a condition that would cause or compromise its efficacy.
Okay, a couple questions about the allergic reaction that to the vaccine. So one person wants to know if it's possible you could have a reaction if you're allergic to penicillin, or you have seasonal allergies or food allergies. And someone else wants to know if having their smallpox vaccination and the scar to show it would have any impact at all on this vaccine.
As far as we know, there is no data to suggest that allergies, food allergies, predispose individuals to a adverse event from the mRNA Pfizer vaccine at this point, or the mRNA vaccine from Moderna. When we talk about individuals who have had a history of substantial immune response to other vaccines previously, that's the main concern around the Pfizer vaccine and holding off on receiving the Pfizer vaccine if you are an individual that falls into that category. Regarding the smallpox, there's no evidence to suggest that is a predisposer to an adverse reaction either to the vaccines in consideration with FDA right now.
Okay. Here's a question that is one of those that could come up that could be controversial in the workplace. Is aborted fetal tissue used in any of the COVID vaccines? What about other additives such as thimerosal, aluminum, and formaldehyde?
Sure. The components of the vaccines are all available in FDA documentation for both Pfizer, and now with the December 17th meeting on the docket, FDA will publish the data on the Moderna vaccine as well, along with some of the non-proprietary items that you speak of. There's no list that I have and can show right now in regards to the full makeup of the vaccine and how it was produced. The proprietary nature of some of that will perhaps not be available to the general public. However, in regards to items that can cause a negative response, such as a formaldehyde, those items will be likely disclosed in the FDA documentation. Again, that's now available in preparation for tomorrow's decision by FDA.
Okay, there's several questions about immunity conferred by the vaccine in comparison to immunity afforded by having had COVID. So I'll just read one of those questions. Can you speculate on how immunity conferred by the anticipated vaccines will compare to immunity conferred by having recovered from COVID? More effective, less effective, longer lasting versus shorter? Others are asking, and we've covered this before, just about immunity in general after you've had COVID and whether you should still take precautions for exposure such as social distancing or wearing a mask.
Sure. So the first part of that question, I don't have to speculate about because we actually have objective data around comparison of natural immunity from COVID-19 and acquired immunity. And the data says they're almost equivalent. And when I mean almost equivalent, we're talking non-significant differences in the response, immune response to the vaccine versus to the virus, native virus itself. So that's positive news. And we're seeing that amongst multiple vaccines in their phase 3 clinical trial. If we're talking about now the latter portion of that question, we have talked about previously 120 days, 90 to 120 days, we see a degradation in the antibodies that are in the immune system still specific to SARS-CoV-2, right? In a different way, we can articulate that the immunity that wanes over time is primarily antibodies. We still see those B cells exist for prolonged periods of time beyond the waxing of the antibodies after natural immunity from COVID-19. It's not studied yet with those who have received vaccinations. Obviously, that data will be forthcoming, but we know that there is hope that the B cells will confer. They're called memory B cells for a reason, because there's hope that those B cells will confer longevity for immunity to COVID-19. However, that's still not yet conclusive.
Okay, we have time for just one more question, and it's regarding the accuracy of testing specific to the new Quest flu and COVID combo test. And another person is just asking about if there's anything new on testing with regard to accuracy that's come out lately that we haven't covered in the past.
So Karen, can you say that again here for me a little louder? It's still cutting out for me.
What is the accuracy of the new COVID flu combo test? And in general, do you know anything new about the testing accuracy in general? If someone's asking, I know we've covered it before, but they just wanted to know if there's anything new regarding which tests are the most accurate.
Sure. So the FDA, there are multiple COVID/flu tests available. The one I presented is novel in that it's for home collection. So that is the huge game changer in terms of getting more tests to more people quickly. The efficacy and accuracy of each of those tests have not changed. So the Quest COVID test still has high accuracy in the 90s for sensitivity and 90s for specificity. Similarly, for flu, those numbers have not changed. Flu actually is a little less in its accuracy in the 80s to 90s, depending on which diagnostic you're looking at, which manufacturer. So we want to talk about then, is there anything new regarding COVID testing diagnostics? And the answer is yes. FDA continues to approve EUAs for both serologic testing as well as diagnostic nucleic acid testing. And there's nearly 300 tests available at this point in time, far too many to articulate all of them, but we'll continue to highlight those which may be game changers for you with regard to safety in your workforce.
Okay, well, we're out of time. We still have some questions left. Just a reminder for those of you who are still with us, we post answers to all questions on our website. So if you look back through that archive, you're going to find a lot of information that we've covered in the past. And it's quite informative, and then we refresh it every week. So check that out, and be sure to listen to the recording. You can see the slides that way. We don't customarily redistribute the PowerPoint. Also watch for a new FAQ we're going to be posting today on quarantine and reducing quarantine from 14 to 10 to 7 days, depending on the circumstances, which is a topic we covered last week. And we're also preparing to publish a vaccine explainer for our clients, which is going to give guidance on how to prepare for distribution, and it has some advice on planning for distribution and some other resources that you can access. So thanks again for joining us. Be safe, take care, and we'll see you next week.
Thanks, everyone. Take care, stay safe, and God bless.